antibodies. And I feel like this is a pretty good graduation from the B cell lecture where we talked about plasma cells that are going to go on to produce these antibodies. So we'll start by talking about just some basics about the antibody structure, some specific regions that you want to be familiar with. We'll talk a little bit about the properties of antibodies, which a lot of this we already kind of touched on, but we'll go into slightly more detail here. And then we'll talk about the different immunoglobulin isotypes that we touched on when we talked about class switching. So the first thing here, when we're talking about antibody structure, really you can organize this pretty simply into two regions. So you have the FC region, and the FC region, as you can see down here, this is going to be kind of the constant region. So that's what the C is. It's standing for fragment constant region. Okay, so the fragments are going to be all of these kind of little squares you see here. And sometimes these squares will get named. You might call this, region 1 and region 2 and region 3, but even when you get to that level of detail, you start to get beyond the scope of step 1. So we just call these regions constant regions. And so again, what we highlighted here in orange is going to represent the heavy chain. Okay, so we have the heavy chain, and then we'll also have a light chain. Okay, in the light chain, you can see, I just remember the light chain, it's lighter. It's not as big as the heavy chain. So the light chain is this area that I'm kind of highlighting in teal here. Okay, so we have our heavy chain, we have our light chain, and then in the middle you can see that occasionally we'll have these disulfide bonds that are going to link these two portions of the antibody together. And so we already said the FC region is kind of like the constant region. Okay, this is kind of like the constant region. That's what that C is standing for. And then the AB here stands for antigen binding. Okay, so this is the antigen binding region, which actually makes a lot of sense. So if you understand some of these terms, you can kind of understand what's going on here. Let's just say, theoretically, we run into some bacteria. Okay, so I'm just going to draw some bacteria here. Okay, so here's some bacteria. And on this bacteria might have an antigen. And it might just so happen that this antigen binds to this portion of the antibody here in the FAB region. And so that's kind of where this name comes from, because the antigen binding will occur in this region. Okay, so the antigen might bind here. And again, there's going to be a very specific antigen that's going to bind to this FAB region, particularly the kind of distal end here of this region. And that's why this is sometimes also called the variable region. Okay, so let's recap some of this just to clean things up. So, there's a few different ways you can categorize this antibody. The first kind of way is we have a heavy chain, which is in the middle here. So this whole thing is your heavy chain. And then we also have a light chain, which is on the outside here. Okay, now the heavy chain you'll see is denoted with this H next to each of these letters here. So you can see there's an H, there's an H, there's an H, and there's different H's, right? This is going to be 1, this is H2, this is H3, right? So there's different ones, but all of these are representative of the heavy chain because it's in the middle. Remember, this is the heavy chain. That's why it gets the H. And then again, the light chain is denoted with the subletter L, right? Because that's the light chain. And then this letter here, there's a V and a C. So the C, again, stands for the constant region, okay? So the C is kind of the constant region, meaning it's going to be consistent more or less throughout similar forms of this antibody, which we'll get to. But there's also a region on the end here that has a V, and so the V stands for the variable region, okay? So the variable region is highlighted here in pink, and again, the variable region is going to be different for different antibodies, and these variable regions will have specific antigens that they can bind to, different shapes, different sizes, right, that these different antibodies can bind to. And the last thing that we talked about is you can split this up into a constant fragment, which is more or less going to be down in this region, and then an antigen binding segment, which is mostly going to be up here. Sometimes you'll see the term epitope used, and this is really just referring to the specific part of the antigen that variable region on the antibody is going to be binding to. So you might say that, this antibody is binding to its epitope, right, or the epitope on this antigen, that specific region that it can kind of match up geometrically to kind of fit into that space molecularly, okay? So that's what we're talking about when we say, that it's binding to its epitope. So the FC region, again, the FC region down here is basically going to consist of our heavy chains, right? So we have some of our heavy chains in the constant region, and this is going to determine the isotype of this antibody. In other words, when we say isotype, we're talking about things like IgM, IgA, IgE, right? All of these different immunoglobulins. So again, isotypes, you have your IgM, IgG, IgE. These are all different isotypes of the antibody. This region doesn't determine what epitope the antibody can bind to, right? That's for the antigen binding region. In the constant region, we're just determining the isotype. So what type of antibody is it? And in this region, we also have the C terminus. And so some people remember this by saying, hey, there's the constant region here, constant is C, and there's also a C terminus. And there's also, since we're talking about C, site of complement binding. IgM and IgG will use this region in particular for binding complement and then activating complement. So if we go back to the scenario where we have some bacteria here, right? So we just have some bacteria here, okay, that's floating around in the blood, and then potentially this bacteria might have an antigen on its surface, or a couple different types of antigens on its surface. Okay, and then what we might have happen is we might have, let's just say we have an antibody kind of floating around here, and it binds to one of those antigens. Okay, and again, it's going to bind using the Fab region, and then we have our constant region down here. Okay, so that's essentially what's happening. Now, once that binds, we can then activate the complement cascade, right? So we can activate the complement cascade, so we can get complement to activate its cascade and eventually lead to a membrane attack complex punching holes through that bacterial cell membrane. Okay, so that's what we're talking about with complement binding. And again, this happens with IgM and IgG. Specifically, the complement will get activated by this portion here, the CH2 constant region. So that is the portion where we will have complement binding. Okay, so I'm just going to write that in here. So complement will bind to that region on the FC portion like we shown here, and that's going to activate the complement cascade to then lead to the membrane attack complex coming in here and punching some holes in this bacteria. Now, the other route we can go here is opsonization, right? So it's kind of a similar concept. So again, if we take our antibody, so again, let's take our antibody binding here. And again, this is going to be the Fab portion. And then we have our FC fragment back here. And again, it's the same concept. If I have, some phagocytic cell around here, so let's just say that I have, let's just say that I have like a neutrophil or some phagocytic cell here. This cell can kind of get roped in to binding to this FC portion. Okay, so it's going to have a receptor here that can bind to this FC portion, and that FC portion will essentially stimulate this phagocytic cell to then undergo phagocytosis and digest this bacteria. Okay, again, binding to the FC portion. And so that's actually going to happen here at this third heavy fragment. This is where we're going to stimulate phagocytosis through opsonization. I'll just put that down here, opsonization. So the idea is if we can mark these bacteria with antibodies, we can have a few things happen. We can stimulate complement binding, we can stimulate opsonization, which will lead to phagocytosis of that bacteria. If we have enough antibodies around, we can essentially neutralize this bacteria or virus, right? We can bind to a lot of these antigen binding sites and prevent this bacteria from going on to reproduce or to infect different cells, whatever this pathogen is. By binding to multiple antigens here, we can essentially neutralize this pathogen. Okay, so the three things we can do: complement activation, opsonization, those are the big two, but we can also neutralize this bacteria by just basically flooding it with antibodies. So that's the tricky part. So in the FC region, we have a couple things going on. Complement activation, opsonization are going to happen at this heavy chain region. The variable region is what's binding the epitope. Remember, that's what's binding the epitope, binds antigens via the epitope, and this variable region will determine the idiotype. So the idiotype is referring to what this antibody can specifically bind to, what specific structure, specific epitope can it bind to, and with what strength can it bind to that epitope. So there's usually going to be a very unique antigen binding pocket, and that's referring to this idiotype. And this idiotype is going to be formed through things like VDJ recombination, which we'll talk about in the next slide. And just to put a plug in here, again, going back to opsonization, remember, what are the two big things that are going to kind of lead to opsonization? So this is particularly high yield. Remember, it's going to be IgG and C3b. Okay, so when you're thinking opsonization, right, when you're thinking phagocytosis, think of the constant heavy region 3, right, this region closest to the C terminus on IgG, and then also keep in mind that complement is going to be, again, like we said, at the CH2 region here.